postdoc is helping. Great, thank you. So hi everyone, welcome to Spatial Omics Seminar Series 6. Today, we're very excited to have Dr. Jeff Spraggins from Vanderbilt University. And briefly, Dr. Spraggins received his PhD in analytical chemistry from University of Delaware. And after that, he joined Vanderbilt University. First, he worked as a postdoctoral scholar, then a research assistant professor. Before starting his job as an assistant professor at the Department of Cell and Developmental Biology, he also started as director of Biomolecular Multimodal Imaging Center, for short Bionic. And his lab has been developing advanced imaging mass spectrometry and integrated multimodal molecular imaging technologies capable of providing a systems biology view of the tissue microenvironment that spans a wide range of molecular classes, metabolites, lipids, proteins, and genes, and spatial scales, single cells, functional tissue units, and whole organs. And they are applying these technologies to various biomedical research areas, including infectious diseases and diabetes. And he has also received numerous awards and grants, and recently he has been playing an instrumental role in Hubmap research program at the NIH. And without further ado, I'd like to turn over to Dr. Spraggins.
Thank you very much for that kind introduction. So as the title suggests, today I'm going to talk about some of the technology developments that we've been working on, both on the instrumentation side of things, developing an imaging mass spectrometry specifically, and then also how we connect imaging mass spectrometry with other forms of biomedical imaging. So as was just said, my research focus is on next generation imaging mass spec technologies. And from an instrumentation side of things, we're working on improving kind of all of the figures of merits for the mass spectrometry based imaging experiments. So things like sensitivity, specificity, spatial resolution, and so forth. I'll talk about one aspect of that work today in how we're starting to utilize gas phase separations using ion mobility spectrometry. I'm also very interested in developing experimental workflows and the computational pipelines that are necessary to combine imaging mass spec with other forms of molecular imaging, as was just stated, to provide more of a systems biology view of the tissue microenvironment and cellular resolution. I think imaging mass spectrometry is a really unique form of spatial omics and molecular imaging because it combines the spatial characteristics of microscopy with the specificity and performance of a mass spectrometer, because we're using the mass spectrometer to generate the ion images, but we're doing it in an untargeted way. So there's no antibody-based tags. So we take advantage of the multiplexed advantage, the specificity advantages of the mass spectrometer to generate images of everything from lipids, metabolites, glycans, peptides, and proteins, the endogenous molecules within the tissue. And it's this combination of kind of deep molecular coverage with a high degree of spatial information that allows it to so well connect the various bioanalytical imaging technologies and analytical technologies. So on one hand, we can make connections with the molecular content to the kind of traditional, deeper LC-based omics technologies, things like LC-based lipidomics, metabolomics, and proteomics, obviously. More recently, we're starting to make connections to spatial transcriptomics. And then the spatial aspect, we can also use that to connect to the traditional imaging modalities, everything from tag microscopy, stain microscopy, and also some in vivo imaging. And when we connect all these things together, we refer to this as multimodal imaging. And again, we can then understand relationships between a wide range of molecular classes, but all the while be able to localize those molecular profiles to specific cell types in functional regions within the tissue. Now, everything I'm going to talk about today is going to be contextualized in terms of one of our application areas. This is our work as part of the Human Biomolecular Atlas program, or Hubmap. And Hubmap is a large scale NIH consortium that's focused on creating foundational, comprehensive molecular and cellular atlases of normal human tissue. So we're not looking at any sort of diseased organs, it's all normal. And there's about 450 researchers in Hubmap right now across 70 institutions, more or less, worldwide. The primary institutions are highlighted here above. And the consortium in general is organized into three primary components, the first of which are the tissue mapping centers, which we're one of. And those tissue mapping centers are responsible for everything from tissue collection to developing the technologies, the assays, the quality control procedures, generating the molecular and cellular data, and creating those atlases, those maps of our specific organ types. We work very closely with the transformative technology development projects and the rapid technology implementation projects. And these groups are developing kind of complementary technologies to what we're doing in the Tissue Mapping Center, or some newer technologies that are kind of pushing the boundaries of what's possible for this kind of mapping. And then all of us work hand in hand with the bioinformatics groups, the component that we call the Hive, or the Hubmap integration, visualization, and engagement component. We're obviously taking all of this data, integrating it, developing the tools so that we can visualize it and share it with the broader research community and the general public. Now, the organs that Hubmap are focused on are shown here on the right, again, looking at normal human organs. Our particular tissue mapping center is focused on the kidney, the pancreas, and the eye. Today, I'm going to focus this entire talk on some of our data for the human kidney. Now, what makes our tissue mapping center unique is this concept of making connections between various types of molecular imaging and spatial omics. And we really strive to do as much as we can with a single tissue section so that we kind of minimize the spatial mismatch between these different modalities, and it allows us to integrate the data more tightly. And we're really leveraging kind of three major things. The first is some of the state-of-the-art imaging mass spec technologies that we're developing at Vanderbilt. We're very lucky at Vanderbilt in that we spatially, localized. We're very close to the Vanderbilt University Medical Center, and we interact with those clinicians very closely. So we have very good clinical partners and pathologists associated with this project. And then, as we were talking about kind of before I got going here, is I work very closely with bioinformatics groups, both here at Vanderbilt as well as collaborators at TU Delft. And the whole concept here is to integrate kind of deep multi-omics, typically collected in a spatially targeted way, with high resolution microscopy, as well as what we call kind of high content imaging, or the imaging mass spectrometry, where we can map hundreds to thousands of biomolecules into single experiment. Now, I do on the very front end want to acknowledge the team, right? So the Tissue Mapping Center that we have at Vanderbilt is kind of actually broken into two grants. The first of which is our kidney group. I just say that the organization of these tissue mapping centers is kind of the leadership, and then we have the organ-specific projects, in this case, the kidney, and then the eye and the pancreas, and then our data analysis core. As I said, we partner with clinicians and pathologists. And on the kidney project, this is Mark DeCastuker, Ray Harris, and Agnes Fogo, and then here's our analytical team. And then you'll hear a lot about some of the work that our data analysis core is doing, and some of the computational tools that they're developing. So we have some folks that are here at Vanderbilt, as well as our partners at TU Delft as well. Just to give a sense of what we mean kind of practically, experimentally by multimodal imaging, in our case, this is just a nice schematic of our kind of entire workflow. And frankly, this is kind of an internal document that we use to help kind of keep track of the metadata and protocols and so forth. The point is not to focus on any of the small text here. That's, I'm just going to kind of use this as a guide to walk through what we're doing with the tissue. The modalities that we're collecting are listed here on the left. And all of our tissue goes through a pipeline that's kind of similar to this. We collect kidney tissue from one of two sources. The first of which are tissue blocks collected from total nephrectomies through the Vanderbilt University Medical Center. So we have partnerships with the surgical units there at Vanderbilt. And when there are kidneys that meet our criteria, then we're able to collect blocks of tissue from those kidneys during a resection. Typically, those are associated with cancer. So we're collecting tissue blocks that are as far away from the site of cancer as possible. Or we're collecting tissue from discarded donor tissues. And so these are through donor procurement services like IAM, where we're getting whole organs and then subdividing that organ into many tissue blocks for analysis. Regardless of where the tissue blocks come from, the first step is to determine their normalcy. So they need to be graded as age-appropriate normal based on histopathology assessment by Agnes Fogo's group. And then once that tissue has been deemed to be normal, it goes through one of two pipelines. I should say it's frozen and then cryosection, and those tissue sections go through one of two pipelines. To the left, we generate autofluorescence microscopy, imaging mass spectrometry, and stain microscopy, all in one piece of tissue, one tissue section. The serial section then goes to the right, where we generate autofluorescence microscopy and highly multiplexed immunofluorescence. Each of the modalities that we're collecting have a very specific purpose. As I already talked about, the imaging mass spectrometry gives us this very untargeted approach to molecular imaging. So here we're just mapping, hundreds to thousands of metabolites or lipids or proteins or whatever. Most of the work within Hubmap has been focused on metabolites and lipids, although we're starting to generate more protein data. This is in collaboration with Neil Kelleher's group at Northwestern, who's also part of Hubmap. But I'm not going to show that. Most of what I'll show today is lipids and metabolites. We have a couple different forms of highly multiplexed immunofluorescence, whether it's generated by codex or more kind of traditional cyclic multiplex immunofluorescence. As I mentioned, we generate autofluorescence microscopy. I'm going to talk about this modality throughout this talk, but we're using it in a couple of very unique ways. First of all, utilizing it to allow for co-registration of all of our data sets together. It's a nice modality that we can collect kind of everywhere because we're not adding stains or anything. It has no effect on the molecular characteristics of the tissue. So we can generate autofluorescence before we do anything else. And that drives some of our co-registration and alignment. We then stain the tissue for kind of general morphology, and then bulk multiomics or spatial multiomics to generate annotation databases, things for us to relate the imaging data to as well. We also have kind of a sub-project within Hubmap to generate average anatomical atlases. So here are just a couple of examples here. I'm not going to talk about this, but this is really interesting. One of the things for Hubmap is that we need to create a common coordinate framework that all of the data can be fit to help with integration across multiple sites and even within blocks from multiple patients. And so these are average atlases made up of, in these cases, roughly 5,000 patient scans from ImageView. So all of the MR and CT images collected through the medical center, we can access as de-identified imaging data sets and use that to computationally create average anatomical atlases and segmented atlases for that purpose. But again, not going to talk about that too much today. So as a starting point, the first thing I want to talk about are some of the developments we're making in terms of the imaging mass spectrometry instrumentation. In particular, how we're improving specificity using a form of ion mobility spectrometry called TIMS, or trapped ion mobility spectrometry. Now, just to make sure we're all on the same page, this is just a schematic of how we perform molecular imaging using a mass spectrometer. There's different ways of doing this. So there's DESI and NanoDESI and LISA and SIMS and all sorts of ways that you can generate images with a mass spectrometer. We primarily use MALDI, or matrix-assisted laser desorption ionization. And this is just the general schematic, particularly for lipids and metabolites. This is how it's done. And you start off with a thin tissue section, just like you would in any microscopy modality. So these are sectioned at 5 to 10 micron thickness cryosections that are thaw-mounted onto glass slides. We can work with FFPE tissues. It's actually not the fixation that's the major problem, it's the paraffin that's a big issue. Paraffin ionizes very well, so it tends to interfere with our mass spec signal. It'll dominate the spectrum and suppress signals from the endogenous molecules. So we actually, in some of our tissues that are more delicate, we'll fix them with formalin, but we'll then embed them in something like gelatin or carboxymethylcellulose, an embedding material that's more compatible with mass spectrometry. The data I'm going to show today, though, is all fresh frozen. Once they're sectioned and mounted onto the glass slide, we then apply a MALDI matrix. This is just a small molecule that assists with desorption and ionization of the endogenous molecules within the tissue. Typically, we're using pneumatic sprayers, and we do that in a way that we're not delocalizing anything. We're not moving the molecules around within the tissue, but we can also use sublimation. Typically, they're kind of drier techniques of applying the matrix. If you overly wet the tissue when you're applying that, you're going to move the molecules around, and then that degrades your spatial resolution. Once the matrix is applied, we then simply go from one pixel to another, firing the laser. generating ions from the endogenous molecules and detecting those ions in the mass spectrometer. So what we end up with are hundreds of thousands of spectra. You can think of each pixel location as its own mass spectrometry experiment. And any of the peaks within the spectra, we can represent as a heat map over that sample area, or what we call an ion image. And so that's the general workflow. If we're doing proteins, the only additional step is basically before we apply the matrix, we're going to go through a series of washes where we wash away the lipids and metabolites that will, outcompete the proteins for charge. And so that improves our performance for protein imaging, but it's effectively the same workflow. This is a schematic of the instrument that I'm going to talk about today, this timsTOF Pro. This is a system that was commercially available by Bruker as an LC-based electrospray ionization instrument. And we wrote an NSF grant with Bruker to develop the MALDI version of this system. And so this is now commercially available. This, all the data I'm going to show here is from the first prototype system that we built with Bruker at Vanderbilt. So the schematic of the system is shown here. So it's the basic mass spectrometer is a dual TIMS funnel. This is an ion funnel that helps guide the ions into the mass spectrometer, but it's specialized in that we have the ability to perform ion mobility here. And I'll talk about that in a second. We have a transfer multipole that just helps guide the ions. We have a mass selecting quadrupole that allows us to do selection, and then we can fragment those selected ions in the collision cell, just like most mass spectrometers. The mass analyzer in this case is an orthogonal reflectron time of flight. So it gets pretty good performance, about 40,000 resolving power and very good mass accuracy, sub 5 ppm mass accuracy. Pretty standard for a orthogonal time of flight based system. Obviously, the unique aspect of the MALDI platform is the integration of the laser and laser optics to allow us to probe the sample using MALDI. This system utilizes what they call the Bruker SmartBeam 3D. This is a 10 kilohertz laser with internal galvo mirrors that allow you to effectively steer the laser beam. What that allows you to do is generate data very rapidly with good sensitivity, because you can effectively etch square pixels by steering the laser beam, while at the same time moving the stage. So this kind of continuous motion and kind of jumping the laser from one pixel to another allows you to create etch discrete pixels with the MALDI experiment, but still kind of maintain continuous motion. So there's some, it's a bit of a dance between the sample stage and the laser to do that. But you can generate data at 20 to 30 pixels per second, and you can etch all of the material within a 10 micron pixel, as I'm showing here. So you're maximizing the sensitivity. You're not leaving any material behind. So you're ionizing as many molecules as possible from a 10 micron pixel. Just to give you a sense of what this system is capable of, just in standard QTOF operation, this is an example from rat kidney tissue. One thing I didn't point out is that I think a lot of people think of MALDI as a destructive technology. And it is true, you are extracting molecules and detecting those molecules. Those specific molecules are gone, but there's plenty of material left behind, and you don't actually etch away the tissue in the process of doing this. You're really just etching away the matrix layer. So the molecules that are extracted in the matrix, you're etching that away. So the tissue is left behind intact. And if you run the experiment right, it's still of good quality. So this is not the best H&E, to be fair, but it is a stained tissue section after the fact. We have a bit of cracking, but that's due to some other things. But the tissue is left behind. We can get other imaging modalities after the imaging mass spec experiment. So what you're looking at here is kind of the bean shape of the kidney, where we have the medulla and the cortex are on the outside. The imaging data I'm going to show you is just about 600,000 pixels at 15 micron spatial resolution. Now, as I said, every pixel is its own mass spec experiment. So here we're looking at just the mean spectrum, the average spectrum over the whole imaging run. And we can zoom in to see how many species we're detecting. So on typical experiments in QTOF mode, we're looking at somewhere between maybe 1,500 to 2,000 peaks that we're detecting. Of course, there is some chemical noise there. There's some isotopes and things like that. So you end up with somewhere on the order of 300 to 800 molecular species that we're detecting in a single experiment. Any of these peaks we can represent as an ion image, and that's what I'm showing here. So on the right is just an overlay of the 3 lipid species that I've highlighted here. These are just false colors. So in pink, we have a sphingomyelin that's localized to the outer medulla. In this cyan color, we have this plasmalogen that's localized to the inner medulla. And then we have this phosphatidylcholine that's localized to the cortex. in kind of the most inner part of the medulla in this case. So really nice spatial resolution. And I think this is a great example of the uniqueness of the distribution of lipids and metabolites within these organ systems. Now, this isn't unique to kidney. We can do this on any tissue type. So this is just kind of a set of examples of imaging data collected on the system. From left to right, we have infected soft tissue. So what you're looking here are lipids that are localized specifically to infected abscesses, in this case within a mouse kidney. We can do whole body imaging of mice and mouse pups. We don't do this often, but when we do this, we're typically looking at drug imaging and looking at kind of concentration of drug metabolites in different organ systems within the mouse models. This is the kidney example I just showed you. This is a rodent brain, and this is human kidney. I'll show you lots of examples of this kind of going forward. So it works across many different tissue types. Basically, if you can thaw mount it onto a glass slide and work with it like you would microscopy, we can work with it with imaging mass spectrometry. Now, one of the challenges, though, with the imaging mass spectrometry experiment is we're analyzing very complex biological mixtures. We're looking at biological tissues. However, we're not able to leverage some of the traditional analytical approaches that are typically used in mass spectrometry to deal with complex biological mixtures. So we can't use things like liquid chromatography or gas chromatography or electrophoresis. In those technologies, you're effectively separating out that complex mixture over time so that you don't overwhelm the mass spectrometer. The challenge, though, is LC or GC or electrophoresis, whatever, takes minutes. An LC gradient might be a 30-minute to an hour gradient, which means you would require 30 minutes to an hour per pixel, which obviously is not practical if you're trying to collect hundreds of thousands of pixels. So instead, we're going to rely on post-ionization technologies, things that occur within the mass spectrometer, within the gas phase after the molecules have been ionized. Now, the primary kind of themes for post-ionization strategies for improving performance are highlighted here. We work on many of these things. The one I'm going to focus today is on ion mobility. And this is analogous with LC. It's just instead of performing that separation before you ionize the molecules, we're going to perform gas phase separations on millisecond timescales within the mass spectrometer. So it's much more compatible with the time constraints of an imaging experiment. In this particular system, as I said, this is a TIMS-based system, a trapped ion mobility spectrometry platform. And the TIMS experiment is done in this funnel region of the instrument. Now, the funnel is divided into kind of an accumulation section and an analysis section. That's just so that we can park some ions and accumulate them while we're doing the scan so we can maximize duty cycle and throughput. So kind of ignore that. It's not critical to understanding the concept, but it just helps us with throughput and things like that. But if we think of just the analysis, effectively what we're doing is across the electrodes in this funnel, we're applying an electric field gradient. So as ions are generated with this laser and they make their way from left to right into the mass spectrometer, they're going to feel a force from that electric field gradient that's going to push them back from whence they came, from right to left. So they're going to feel that force pushing them to the left in the way this schematic is set up. At the same time, however, there is gas flowing through this cell from left to right. So they're also going to feel the force from the collisions of that gas pushing them from left to right. So what happens is that the place within this funnel where the force from the gas collisions equals the force from the electric field gradient, the ions are going to park themselves there. And that position is going to be dictated by the size, the shape, the charge of that molecule. And so each of the ions will separate themselves within this TIMS funnel. And then we're simply going to lower that electric field gradient and elute the molecules based on their mobility. So the larger ions that are feeling a greater force from the collisions are going to elute first, and the smaller ions will elute last. Now, importantly, what's unique about TIMS relative to other forms of ion mobility is that we have control over the performance. So depending on how fast we scan this electric field gradient, that'll determine how much separation we can get in the gas phase. So we can operate at a kind of low resolution, which is similar to many other types of ion mobility, around 50 resolving power, or we can scan more slowly and get upwards of 200 or 300 resolving power with this platform. So it's very flexible, which is nice for the imaging applications. So just an example of what this system is capable of. This is some data collected from human kidney. These are some of our Hubmap samples. Excuse me. On the right there is an autofluorescence image collected before the analysis is done. The gold box here is where the data that I'm going to show you comes from. So we have a bit of medulla on the left and some cortex on the right. Now, this is all done on this prototype system, about a 400 millisecond scan time. Of course, to get like the highest performance, you're going to slow down your acquisition rate. So it's a slower experiment. It's about 2 to 3 pixels per second in this case, but you get more performance out of it. Similar to the experiment I showed you previously, we can just look at the spectral data, and this is what you get. It's a mean spectrum. We can zoom in thousands of peaks within this. And of course, you can represent these as ion images. So if we just look at the mass to charge ratio dimension, mass versus intensity, we end up with, after some processing, getting rid of some of the noise and de-isotoping and so forth, we end up with about 680 molecular features, which is quite good. It's pretty standard for an imaging mass spectrometry experiment. And even with mobility kind of activated, although we're not using it, we still get really good sensitivity and spatial resolution. So here's just an example of 4 different ions, a couple of single myelins, a couple of phosphatidylcholines that have nice localization. Again, left is the medulla, right is the cortex. So for instance, this particular single myelin localizes to the glomeruli within the cortex, whereas this one localizes to the proximal tubules. So we get really good data quality. But the thing to keep in mind is we have this extra dimension of information. Aside from the standard mass to charge ratio and intensity, we also have ion mobility. And one way to look at this is in this mobility heat map that I'm showing here. You have mass to charge ratio along the x-axis and mobility along the y-axis. The intensity then is encoded in the color in this case. Now, this is a nice way to kind of demonstrate that you have this extra dimension of information, but it's a little bit hard to look at. I kind of like this version's a little bit more interpretable. So we have mass to charge ratio along this axis. We have ion mobility along this. And then now we have intensity and just the height of these extracted ion mobilograms for each of these mass to charge ratios. And what you can make out if you start to look at this closely as it kind of rotates around is that some of the peaks, like this first one, is just a single peak. So there's one species at this mass to charge ratio, but many of them have multiple peaks. And so what that suggests is that we're separating out in the gas phase molecules that have the same or very similar mass to charge ratios, and that are probably either very close isobars or in some cases true isomers, things that have the exact same molecular weight that we can now separate with ion mobility. And analytically speaking, this gives us a big boost in a lot of our figures of merit, things like signal to noise ratio and just overall molecular coverage. So now if we take into account this ion mobility dimension, we go from 680 molecular features to over 2,000 molecular features after we do some processing. And we can look at just one example just to highlight the power of this technology. And so what you're looking at here are 3 panels, where in each panel we have the respective ion images above. And below on the bottom left, we have the mass spectrum. In each case, we've zoomed in very far on this. So this is 756.2 on the left side of this and 756.8. So it's a 0.6 Dalton window in mass spectrum space and M over Z space. And so it's just to point out that we're looking at one peak in the mass spectrum. Now, each peak within our mass spectrum, we can then generate an extracted ion mobilogram. So this is what the mobility data looks like. And we see that we can detect multiple peaks here. And then, depending on the window that we selected for this mobilogram, we generate the ion image. So on the left, we've effectively selected the whole mobilogram. This is what the image would look like, effectively if you didn't have ion mobility. You're just kind of averaging it all out. Again, on the left is the medulla, on the right is the cortex of this image. Now, importantly, we've been able to go in and identify these things. So this is a phosphatidyl 34,3. With the mobility information, we can figure out that this peak on the left is a phosphatidylcholine 16,1,18,2. What these numbers represent are the numbers of carbons and double bonds within the acyl chain of the fatty acid of these phospholipids. So one of the fatty acid chains has 16 carbons and 1 double bond, the other has 18 carbons and 2 double bonds. And we see the localization of this molecule here. And then the peak on the right is a PC 16,0,18,3. So what we're looking at is now the ability to differentiate molecules that are only different in the position of one of their double bonds. These are true isomers, they have the exact same molecular weight. It's just a structural difference between where the double bond is within the fatty acid tails. Not only can we differentiate those things in the gas phase with ion mobility, but with the imaging experiment, we can see that they actually localize to quite different regions, particularly within the cortex of the kidney, the area to the right here. So this particular ion here kind of localizes to the majority of the cortex other than the glomeruli. These black spots are the glomeruli. Whereas in pink, we see that this has a much more sparse distribution. This is actually localizing more to the distal tubules than the proximal tubules, and it's also absent in the glomeruli as well. So you have different structural localizations within the tissue with very minor structural difference within the molecule itself. So that's some of the technology that we're developing. Now I want to shift gears and talk about this concept of multimodal imaging, how we're combining imaging mass spec with other forms of molecular imaging and imaging in general. And so as a starting point, just to highlight what the data look like that we generate for Hubmap in this kind of multimodal approach, this is an example data set from a 38-year-old female donor. This is the autofluorescence shown here on the right. So we have cortex, a bit of medulla. The image regions that I'm going to show you are from this white box here. Now, this is the area where we collected the imaging mass spectrometry. All of the microscopy is collected by using whole slide imaging. I'm just not going to show all of that. I'm just going to show the bit from the box here. So this is what it looks like. To the left of the white line, all of that is collected on a single tissue section. So we take a single 7-micron cryosection of the tissue. We generate the autofluorescence microscopy imaging mass spec. I'm just showing 3 molecules here. Of course, there's many more. And then we can stain the tissue after the fact. This is the stained microscopy image. To the right of the white line is the serial section where we generate autofluorescence. It's just redundant, so I didn't show it, as well as multiplexed immunofluorescence. So this is what the whole data set looks like. Now, one of the things I want to talk a little bit about is if we're going to combine these data sets together, we need to think about spatial co-registration. And that's actually quite a challenge in this case. Not only are we dealing with different modalities, we're dealing with modalities that have very different spatial resolutions when we're trying to connect microscopy to imaging mass spectrometry. Typically, the imaging mass spec is about 10 times less resolved than the microscopy, or more in some cases. And so that introduces some real challenges. So we're dealing with the standard kind of rotational warping effects, but then we have to figure out how to deal with this mismatch in spatial resolution between the two. And this work is really driven by Heath Patterson in our group. He's developed a set of experimental workflows and computational pipelines that he calls IMS Microlink to make these spatial connections between these data sets. The way this works is shown here, and I'll show a video. This is a bit of a complex figure, but I'll show a video in a second. But effectively what we do is we collect autofluorescence microscopy first. We then generate the imaging mass spectrometry data set. We're going to go back and collect what we call post-acquisition autofluorescence, this image here in gray. Now, importantly, with that post-acquisition autofluorescence image, we're not going to wash away the matrix. So we're going to leave the matrix there. And what that allows us to do is visualize the craters. So again, we shouldn't be punching holes in the tissue if we've set up the image correctly. We should only be kind of ablating the matrix material. But if we leave the matrix behind, we can see those craters in the matrix. We can create a virtual pixel map. So we now know the exact location of every MALDI pixel and how that relates to the microscopy. So we can use that virtual pixel map to then project the imaging mass spec data into the coordination space of the microscopy. And this is what this looks like. So what I'm going to show here are just a set of images. We're going to scan through some of the microscopy modalities into the imaging mass spec just to highlight the co-registration of these things, just to highlight the accuracy and precision of that. So we're going with the same microscopy into the autofluorescence microscopy. All of this is collected at the same spatial resolution, so we can use kind of morphological features to co-register those. We then have this post-acquisition autofluorescence scan where we can make out the grid of pixels that we've collected as part of the imaging mass spec experiment. The software automatically determines the centroids of those. And so we now have this virtual pixel map that we can project the imaging mass spectrometry data into. Now, this is kind of an exaggerated case because this is collected at 30 micron spatial resolution. All of our Hubmap data is collected at 10 micron spatial resolution. But even for low spatial resolution imaging mass spec data, this process works. Now, aside from co-registration of the data, we're also starting to utilize the autofluorescence for data mining. And so the idea here is that we want to utilize morphological information that's embedded and coded within the autofluorescence for specific functional units of the kidney in this case. We want to create segmentation masks using the autofluorescence so that we can mine the imaging mass spectrometry data in biologically informative ways. And the reason why we want to do this is, obviously the autofluorescence, just like we like using it for co-registration, the benefit of using autofluorescence is that we don't need any stains or anything like that. So we can create models that can interpret these autofluorescence images it gives us a way to create these important segmentation masks without having to add any stains or do anything that might make the tissue incompatible with imaging mass spec. And so the approach here is to use these fluorescence images. We're also going to compare them directly to PIS stains. This is work done by Heath Patterson, I should point out, within the group. And Heath uses convolutional neural networks, a pretty standard approach for creating these segmentation masks. Now the workflow goes something like this. And this is on bioRxiv. I should have put the reference here, but we co-register the autofluorescence microscopy and the PIS stain microscopy. Once they're co-registered, they are annotated by Agnes Fogo and our pathologists that are part of our Hubmap project. We then tile these whole slide images. Importantly, the tiles are matched between the PIS and the autofluorescence, and then we build out our training sets. Now for this experiment, we're interested in comparing performance between kind of classical stain microscopy, which is often used for this purpose, with performance of autofluorescence. So we're going to specifically match those random training sets between the PIS and the autofluorescence. So we're comparing apples to apples. We're then going to use CNNs to build a model that can predict, in this case, glomeruli, but we're also doing this for all of the functional units within the kidney. And then in a pretty standard way, we're going to build models with a portion of the data set. We're going to hold a portion of the data set back to do some testing and assessment using things like true positive rate and dice source and coefficients and so forth. This is what some of those results look like. So here you're looking at true positive rate as a function of the number of glomeruli we've used to train the models. On the left is the data, on the left is the data for the FFPE tissue. On the right is fresh frozen, so we looked at it in both cases. And in both cases, the PIS stain images are in pink and the autofluorescence is in this kind of bluish color. And what we see is that the performance is equally good for both stain tissue and autofluorescence, just demonstrating that the autofluorescence works perfectly well for this kind of functional tissue unit level segmentation. And in both cases, if you throw a couple hundred glomeruli at it, they're going to perform very well. And in this case, segmenting glomeruli, but again, we can do this for proximal tubules, distal tubules, collecting ducts, vasculature, all of the primary structures within the kidney. Now, the reason why this is so important, we want to develop these automated strategies for segmenting the structures within the kidney, because in Hubmap, we need to operate at scale. So even within just our first data release, where we have 24 patients samples, we're doing whole slide images. The autofluorescence images for all 24 samples are shown here on the right. And in these cases, we've automatically segmented the glomeruli. They're a little hard to make out in these whole slide images, so I've just zoomed in on a few here. You can see the segments highlighted in this kind of yellow to red gradient. Just within these 24 samples, there's over 5,000 glomeruli that we've segmented. Now that's hard enough, but the glomeruli are kind of the low-hanging fruit. There's just fewer of them than the other structures within the kidney. It's even more critical when we think about things like proximal tubules. So we look at just one tissue block and we're looking at segmenting proximal tubules. Now, to be fair, this particular tissue block is all cortex, which is where we find the proximal tubules. Just within this one tissue section, there's 11,000 proximal tubules that we can now segment with the autofluorescence microscopy. Across the 24 patient samples, now we're talking about 250,000 proximal tubules. Obviously not practical without these kinds of tools. Now, what I'm not showing here today is we do have matched multiplex immunofluorescence data that we've used to confirm these results and used to validate the segmentation pipelines, but I don't have time to get into that today. Now, what do we want to do with this? So we have imaging mass spectrometry. We've got a couple different forms of microscopy. We're using that microscopy to now segment functional tissue units within the kidney. We're going to utilize supervised machine learning and classification modeling to mine these data. So when we think about classification modeling for imaging mass spectrometry, the standard workflow is highlighted here. So we're using a mass spectrometer to generate pixels within our images. Each of those pixels, a researcher is going to come along and label them. So in the case of the kidney, they might label a pixel as being a part of the cortex or the glomeruli or medulla or proximal tubules or distal tubules or whatever. And once you have that labeled data, you're going to then use a supervised machine learning algorithm to create a classification model. Pretty standard. Now, in our case, we're not going to let a researcher do that labeling process. We're going to utilize the autofluorescence microscopy because we can do that at scale. We can do that automatically. But the whole point is to effectively label the pixels for what functional tissue unit they are associated with. Now, with a normal classification task, usually what you're going to do then is come along with new spectra that are unlabeled, that have not been designated to be one of these structures, and you're going to use that classification model to label the data as being one of your classification groups. That's not what we're going to do. So we're not necessarily interested in this part of it. Instead, what we want to do is understand how the model learned to make the call. How did the model learn to say that this pixel is associated with the glomeruli or associated with the medulla or the cortex or the proximal tubule or whatever? And so we're going to utilize interpretable machine learning or Shapley additive explanations. And this is all driven by Rong Fan and Prof at TU Delft and his student, Leonore. And SHAP is really interesting because it gives us effectively a quantitative measure of ranking. It allows us to kind of quantitate the importance of each molecular feature, its importance in how the model made its decision for whether or not a pixel is associated with the glomeruli, for instance. And so effectively we get a SHAP number associated with each molecule, associated with each feature, and we can then just create a ranked list of potential markers for a feature based on its predictive performance in order. And so what it allows us to do is take data across many patients. We build a high quality classification model. We can then open up that model, look at how the computer made its decision, and determine the molecular markers associated with those structures based on their predictive performance in this case. Now, the reason why this is so important is because the data sets get immense. So even if we just go back to this first data set that's just 24 patients, this is now kind of a mosaic of mosaics, if you will. So within this data set, each of these little thumbnails are individual ion images. And just within these 24 patient data, one of the things I didn't point out is we generate both positive ion mode and negative ion mode data. So we have these two modalities because they generate images for different classes of molecules. But we end up with 48 imaging mass spec data sets, which after some processing, we start off with about 14 million mass spectra, but after some processing, we end up with 20,000 molecular images. So it's just a lot of data. How do you interpret that? On the biology side of things, we have, as I said, thousands of glomeruli, proximal tubules, and distal tubules. And so now this gives us the ability to take that data and automatically distill it down to the most important molecular features, a molecular profile for those specific functional tissue units. So that's what you're looking at here. So here are plots for the individual lipids that we've determined to be molecular markers for the glomerulus. These are their global SHAP values. So this is how we're making this call as to what is a marker and what's not a marker. The positive data is highlighted here on the left. The negative data is highlighted here on the right. And of course, now that we've got this ability to do this, we can, as a sanity check, we can go look at the images individually. And so this is just an ion image of a 48-year-old white male and a 44-year-old white female for our #1 marker for glomeruli within these human patients. I should point out that the color scale is inverted here. So darker is higher intensity. And so what you see is that this particular molecule across all of our patient samples, we've confirmed this, is higher intensity within our glomeruli. So now this gives us the ability to automatically filter in this way. Now, interestingly, we're still working out the biological importance of all these species, but this particular species is one that's been identified previously as being important for the glomerulus. It seems to be associated with the podocytes, and it's important in progression of diabetes. So it's nice to see that we've done this using machine learning in an untargeted way. We're seeing results that are backed up by the literature in this case. Now, the last thing I want to spend just the last few minutes on is how we're going from 2-dimensional imaging to 3-dimensional imaging. And this will be really quick, but basically what we do is we look at this co-registration of 2-dimensional data. We're just going to expand into 3D. So we're going to utilize the autofluorescence not only to register one modality to another, but also to create our 3-dimensional volume. So we have this monomodal approach to lining up serial sections, hundreds of serial sections across a 3-dimensional block. This is just an example of just the autofluorescence data, a 3-dimensional volume. This is just a little test case of 23 sections. And then this is just kind of scanning in and out of the medulla, in this case, looking at the various tubules. This, again, is just autofluorescence data that we're showing here. This isn't anything else. Now, just as an example of what we can do, this is that same 38-year-old patient. This was a little pilot study doing 3-dimensional imaging generating autofluorescence, imaging mass spec, stain microscopy, and multiplex immunofluorescence. I'm just going to show the imaging mass spec data, the autofluorescence, and the stain microscopy in this case from this region here. And these are what the volumes look like. So using the autofluorescence, not only can we co-register data with high accuracy and precision within a two-dimensional plane, but now we can generate higher spatial resolution, three-dimensional volumes for imaging mass spectrometry data as well. So with that, to conclude, what I hope to show you today are some methods that we're developing to improve our specificity, to generally improve imaging performance and in terms of instrumentation development. Also, some of our unique workflows for combining imaging mass spectrometry with other forms of microscopy and molecular imaging to really get at the molecular drivers of biology in a localized way, understanding the molecular profiles of specific functional tissue units, but we're also moving towards utilizing the multiplex immunofluorescence data more to start linking those profiles to specific cell types as well. Obviously, we talked about some of the computational pipelines, some of the experimental workflows, and there's a lot of people to thank in a project like this, and those folks are highlighted here. So the VU Bionic team that I highlighted earlier are here in the middle. I also want to note Katerina and Emilio. They are two wonderful graduate students in the group that are working on some of the ion mobility development, our funding, and our wonderful collaborators both within Hubmap and across Vanderbilt University. So with that, thank you, and I'll take any questions.
Thank you, Jeff, for this fantastic presentation. Very cool 2D and a 3D chemical literal world inside our tissue that we didn't know. Thank you for sharing.
Yeah.
So if there are any questions, please feel free to post on the chat or unmute yourself and ask directly to Dr. Spraggins. And as we are waiting, maybe I can ask a few questions. So the species that you're looking with the peaks, how definitely are they correspond to certain chemicals? Like what is the confidence for each experiment?
Yeah, it's a good question. So the accuracy, well, it's a combination of how we prepare the sample and the mass accuracy experiment. You know, we're fairly confident, I can't quantitate this, but we know that it's either a metabolite or a lipid, and then based on the mass accuracy, we can narrow down to a specific class of lipids. So we know it's a phosphatidylcholine, we know it has a certain number of carbons and double bonds based on the mass accuracy alone. What we don't know is beyond that, you know, we need to go a little bit further. So I showed that example using the ion mobility data. What I didn't show there is we actually did a lot of fragmentation to make sure we know exactly what those things are from tissue. For Hubmap, what we're doing is we generate a couple of different complementary data sets. We're doing LC lipidomics or LC metabolomics to kind of create an identification database. And then in specialized cases, like those ones that are highlighted in that rolled out of the SHAP process, those we go back and fragment directly from tissue as well to confirm. So we're currently working on a paper for that data. And each of those we go back and fragment directly from the tissue. So we're pretty confident in those. Now, of course, there's kind of levels of validation, right? And so for lipids, for instance, we can tell the localization of, we can tell the structure in terms of the number of carbons and double bonds in the two tails. But with the fragmentation methods that we're utilizing, we really can't tell the location of the double bond along those tails. So we don't get efficient fragmentation of the double bond.
Okay. And there's another question on the chat. Have you compared the result from fresh tissue versus FFPE? How
Sure.
much discordant they are in general? And Dr. Nigedaman is asking.
Yeah, so that's a tricky, well, I shouldn't say it's tricky. It's fairly straightforward to answer. It's a complex answer because it is analyte dependent. And so certain classes of molecules, obviously things with primary means, are going to be modified with the fixation process. So if we're talking about lipids, for instance, the phosphatidylethanolamines are effectively lost in the FFPE in the fixation process. So that class of molecules is just no longer available to you when you do the imaging. The other class of molecules are actually fine. They're okay. Now, the fixation process also delocalizes small metabolites. So things that will solubilize and, during the fixation process will also move around on you. So you would not want to do small metabolite imaging on FFPE tissue because you've lost that spatial information. What's interesting though is FFPE tissues work better for some molecular classes. So for instance, glycans, you can apply enzymes to cleave off N-glycans and go look at the glycans. We're not sure why, but that process works much better in FFPE tissues than it does in fresh frozen tissues. I mean, it's night and day. And so if you see in the literature, there's groups that do this, oh gosh, I'm drawing a blank on his name right now from MUSC. Anyways, there's groups that have specialized in this process. And you'll notice in the literature that they're always using FFPE tissue, and we think this is why, because the fresh frozen just doesn't work very well compared to FFPE. Richard Drake, thank you. Or just drop that in there. Appreciate that.
And how about the morphology of the tissues? Are they the same? Morphology?
Yeah, that's a really good question. So we spent a lot of time thinking about how to freeze fresh frozen tissue to maintain good morphology. So if you just do standard like liquid nitrogen based freezing, it's a little bit too fast and you get lots of cracking and distortion of the proximal tubules. All the proximal tubules kind of collapse. It doesn't look quite as good. Molecularly, it's fine, but at the resolutions we're talking about, everything looks kind of fuzzy because you've collapsed all the tubules. And in particular, if you go try to do multiplex immunofluorescence on those samples, the quality is noticeable. However, if you slow freeze them in a slurry of isopentane and dry ice, that slow freezing process dramatically improves the quality of the tissue section. And there's also things that we do in terms of embedding that help that as well. For instance, like we're starting to transition to fish gelatin as an embedding material that maintains the structure of the tissue even better than what we've done previously with carboxymelted cellulose.
I see. Thank you. And there's another question. What is your opinion in using Spec for phosphorylation pathway? Yeah,
no, of course. The nice thing about what we do is you're detecting the endogenous molecules. So if there is post-translational modifications that are important, we will pick those up in the mass shifts than what we observe. So that's a strength of what we do. Now, the only caveat there, I presume what we're talking about is protein imaging. The only caveat is that our coverage isn't as good. So full disclosure, right? We're not detecting thousands of proteins in a protein imaging experiment. We're detecting hundreds of proteins. So on the order of, depending on the mass spectrometer, it's going to be between 100 and 500 proteins that we're detecting. Now, what's interesting is that we will detect the higher intensity proteoforms. I don't want to say all the proteoforms, but we will detect many proteoforms for a given protein. And in some cases, those have unique localizations. So that is an advantage of imaging in the way that we image versus antibody-based imaging, which can be a trickier thing to get at.
And as we are waiting, and please feel free to unmute if you have any questions. Oh, we have a question actually from Joseph Beecham, yeah. Please.
Hey, hey Jeff, fantastic talk.
Thank you.
I'm curious about lipid signaling is really important in several systems. And what do you find? Can you follow interesting lipid signalings? I'm thinking mainly from the immunological perspective. There's a lot of very interesting phospholipid signaling that almost no one pays attention to. What do you find there?
I know, and honestly, we're just going down this path with this project. I think this is one of the advantages of Hubmap and kind of forcing us to think about the biology more than just I talked a lot about the technology, but we're working towards helping with the interpretation. Like that's the whole point of the multimodal thing, is how do we interpret these data? And so I can't say anything specific right now because we're just starting to go down that road. But I think that's the next step. Like the community now we can all generate really nice lipid images. Now we got to make sense of that. And clearly there's important biochemistry going on because we have such unique distributions. We have kind of classes of molecules that are behaving together in unison within these structures. And so now we're reaching out to our lipid biochemistry friends to help us with that interpretation.
Oh, way cool. Very neat. I'll look forward to seeing that. Thank you.
Thanks. Yeah.
So actually what would be cool is to reconstruct these KEGG pathways with the images directly, right?
Yeah.
Even cell by cell, I think that would be like very cool. And I teach this in systems biology course, and they're pretty handy.
Right.
I mean, I'd like to show you some images in the future.
Yeah, that'd be cool. That'd be great.
Exactly. And any other questions? So it's almost 3. And if there are no other questions, let's thank to our speaker again. Thank you, Dr. Spraggins. I'm not going to use this clapping tool, and we appreciated this fantastic talk.
No, I appreciate it. Thanks for the invitation. And anybody who's interested, just drop me an email if you have other questions. No problem.
Of course. And before we wrap up today, I'd like to also mention that next week we will host Spatial Genomics from Pasadena, California. And the speaker will be Brian Fritz, the marketing manager, and they will be presenting us the advancing biology with spatial genomics. And as you guys know, this is the commercialization effort of sequential fish. And we look forward to seeing you next week. And with this, then we are wrapping up for today. Thank you guys, and thanks for participating.
Thanks very much.
Bye-bye for now.